Hi there Year 9 and welcome to your last lesson on your human transport topic. So we've looked at the structure of the heart. We've gone to move on to look at the different blood vessels, the composition of the blood. And last week, we looked at heart rate and heart disease. And some of you may have done a practical in the last week. This is our final topic, so it's the final section of your booklet that you've got on OneNote. And this is all on your immune response. So linking kind of mainly back in with the composition of the blood. We're going to be looking particularly at white blood cells. So you can see there's a starter task up on the screen right now. So if you want to pause this video, list as many ways you can think of that our bodies defend themselves before a pathogen enters our body. So actually before the pathogen enters our body, whatever pathogen we're thinking about, whether it be coronavirus, whether it's the flu, what do our bodies do to actually stop it getting in the first place? And we call these non-specific responses. So if you want to pause the video now, have a go at writing down as many as you can think of. So in today's lesson, we're going to be looking at, you can see the stretch challenge and super challenge. We're going to be looking at explaining the role of the immune system. So what our immune system does in order to try and keep us healthy. We're going to explain the immune system response, so actually how it does it, and then we're going to end by looking at the importance of vaccination, so things that we can do. And we're going to try and make this as topical as possible in the current climate as well. You can also see those keywords down the side: phagocyte, lymphocyte, antigen and antitoxin. Hopefully by the end of this session, these keywords, you'll be able to come back to them and you'd actually be able to write a definition for each. So hopefully in your starter, you managed to have a go at maybe coming up with some of these. So our first line of defence, we call it, the first line of defence that stops pathogens getting into our bodies. So we've got in our eyes, our tears produce enzymes that help to kill the bacteria on our skin, believe it or not. That horrible picture there of the ears. So earwax can trap the microbes to prevent them getting in. And in our mouth and our stomachs, you might remember this from digestion topic, the saliva in the mouth and hydrochloric acid in the stomach will help to kill the bacteria too. So all of these are non-specific. So then what that means, and you'll talk about this in your live lesson, is they're not specific to a particular type of pathogen. These defend against all types of pathogens that are trying to enter our body. Continuing over, there are a few more, and you may have come up with some of these. In our nose, in our windpipe, our trachea and lungs. Mucus traps microbes, and the cilia, the small little hairs lining our trachea, help to waft it up towards our throat. So actually, if that pathogen manages to get into our trachea, there'll be little cilia hairs and mucus that can trap the dirt, the pathogens, and help to waft them out before they reach our lungs. And quite simply, your skin, your biggest organ, your skin, secretes antimicrobial compounds, so it can help to kill off the pathogens before they enter, and scabs as well, prevent pathogens getting in at wound sites. And if you think back to the composition of the blood, you might have mentioned platelets making up the blood, and they help the blood to clot and create this scab, to create the barrier for pathogens getting in. So linking back to our previous topic. So all of these things mentioned so far, these are our first line of defence. And you'll see a table a little bit like, well, exactly like this one in your booklets. And you can see our first line of defence. And what I'd like you to do, whether you do it now or you can do it after this, is you can come up with your own little summary in that box. So for all of those things we just ran through, if I show you an example here, just do a little summary talking about this first line of defence that prevents the entry of pathogens into the body. And as you notice here in the middle, it is a non-specific response. So moving on to look at our second line of defence. So the first line of defence hasn't quite worked, and actually a pathogen has entered our bodies. What is going to happen? This is when our immune system will kick in. Okay, and the immune system, this is a generic white blood cell, this thing in the middle, and there are three different types of white blood cells we're going to look at today. Some white blood cells, as you can see here, produce antitoxins. We'll talk about those more in a minute. Some white blood cells carry out something called phagocytosis, and some we call them lymphocytes, they produce antibodies, and that's one of our keywords linking back to that starter slide that we're going to go over. So just listen into the next few slides, and you can always go back and use this again when completing your booklet. So we'll start with phagocytosis. So about 70% of the white blood cells are phagocytes. This is part of the body's immune system. They do not produce antibodies like lymphocytes. Instead, as you can see from this picture, they ingest and destroy pathogens like bacteria. What happens when they do this, and you can kind of see this through the process of A and B here, the phagocyte surrounds the bacterial cell, enclosing it into its vacuole, and the enzymes are secreted in the vacuole and they help to break down, they destroy the bacterial cell. So it engulfs, it moves around the bacteria, and as you can see the key word here, it engulfs the bacteria and then it actually destroys it by producing its own enzymes. Now we call this, when you go back to that table, this is still a non-specific response. So a phagocyte can actually do this to multiple different types of pathogen. It's not specific to one pathogen. So this is still classed under it's a second line of defence, but it's a non-specific response. And hopefully in your live lessons you can talk a bit more about that. So as I mentioned, lymphocytes. Lymphocytes are white blood cells that can produce antibodies. So if you look at this picture first, this is the one you'll have in your booklets too, you can see these white blood cells here are producing these antibodies, which are specific to what we call the antigens on the pathogen. And I'll explain that a bit more. So the pathogen here, if we go down to this picture here, this is kind of zooming in on what's happening here. The pathogen here will have specific antigens. Let's change my colour. Will have specific antigens on its surface, and they're specific shapes for that pathogen. So we've got something that's invaded the body and it's got these specific antigens on its surface. And the antibodies, and that's this green one here, which are produced by my white blood cell, by that white blood cell there, they stick to the antigen on the pathogen. So they're specific. This is a specific response. They've got to be exactly the right shape to fit with the antigen on the pathogen. We're going to come back to these two keywords because they sound quite similar, don't they? But we're going to come back to this antibody and antigen later on. What they do when they stick to the antigen is they can cause the pathogens to clump together. And if these pathogens then clump together, the phagocyte, which we've just spoken about, can come along and engulf them all in one, much more easy. And they can also cause the pathogens to burst. So this is a specific line of defence to destroy particular pathogens, because the antibodies need to be the correct shape to bind with the antigen. And like I said, it can either cause them to clump together, so the phagocyte can come along and engulf them, or it can cause the pathogens to burst. And final one, don't worry, it's actually very, very similar. Some pathogens will produce toxins, which can make you feel quite ill. So lymphocytes, as well as producing antibodies, they can produce antitoxins. And these antitoxins will neutralise the toxins produced by the pathogens. So both the antibodies and the antitoxins are highly specific to the antigen on the pathogen. So we call this is still a specific response. So you can see here, my microorganism, this is my pathogen that's invaded the body, produces toxins that might make us feel unwell. The white blood cell, this is the lymphocyte here, produces the antitoxins, and they're specific. You can see their shapes are specific, so they can, I think a good word to use is they can neutralise the toxins, not making you feel as unwell. And that's another specific response because they need to be the right shape. It is good just to mention some misconceptions that can happen in examinations. Sometimes teachers will explain this as a brilliant topic, and you can explain it thinking about a lot of metaphors, invaders and battles. Really try and steer away from that in your exam. Just stick to the point, try and keep as many key scientific words in there. It's good to note the pathogens are not the disease. They cause the disease. So the pathogen entering your body isn't a disease, but it can cause you to get a disease. The white blood cells do not eat the pathogens. So when you talk about phagocytosis, although it looks like they're eating them, don't use that word eat. Try and use the word ingest. And antibodies and antitoxins, the last two mentioned, they're not living things. They're just specialised proteins. So our white blood cells make specialised proteins, antibodies and antitoxins. They are not living things themselves. Those are just key kind of misconceptions that can come up in examinations. So in your booklet, after this, you'll see you've got 3 summaries: phagocytosis, antibody production and antitoxin production. Don't worry too much about the number of boxes I've given you. Just try to make it work best for you. So use the teaching I've done through this, have a look in your textbook and have a go at trying to summarise what is happening. And it might be that it's one simple sentence in each box, or even a key word. So for example, in this box here, this one is just the bacteria. So you might just write bacteria in that box, and the bacteria you can see here in this box produces antitoxins. So it doesn't have to be, don't worry if you think, oh, I'm not filling the whole box. I've had students like that before. So this top box here is just the bacteria and it's producing toxins. So what I'd like you to do is to summarise these 3. Then you'll see there's a 6-mark question. Explain as fully as you can how white blood cells protect us from disease. All you're doing for this 6-marker here is you are bringing together these 3 diagrams, really. So I would aim to talk, maybe briefly explain phagocytosis, antibody production and antitoxin production. And that's roughly 2 marks for each, should get you your 6. If you do finish that, do think about question 2 in this extension box as well. Explain why the body is ready to fight a reoccurring infection. We're going to talk about this next. So actually, once you've been infected with something, why, if you're infected again with it, can the body fight it a bit quicker? And that moves us on really to our next, our final learning aim for today's lesson. In your booklet, you'll also have this. We kind of saw this at the start. Do remember to go back and just make sure you do a nice summary of your learning. And that's quite good for, your revision. A few things to think ahead before your next live lesson. Let's see if I can go back. There we go. I'm not going to run through these now, but if you'd like to pause the video and just have a go at answering some of these are some of the questions that your teachers might ask you in your next live lesson, linking back to this video. These are some quite quick answer ones, just to check you've got the core knowledge. And these are some slightly, there you go, you can see the full thing there. These are some slightly bigger ones, some bigger think questions, particularly your challenge question there. And these are something I'm going to be discussing with my class later on in the week. So if you'd like to pause the video, you might want to just take a time to think about these. So, as I said, our final point. So we're going to think about immunity now. And that's kind of what we just mentioned there in one of those questions. Why, when you're reinfected, can your body fight it off quicker? Something we call immunity. So immunity is when you have the correct antibodies to stop you from getting an infection. Your body already has made these antibodies. So we're going to look at the primary and secondary immune response. Don't get this confused with your first and second line defence. This is different. What it means is the primary response is when you first get infected with a pathogen. The secondary immune response is if you get infected later on, again, with the same pathogen, with the same antigens on its surface. In your booklet, you'll see you've got this summary. When you see summaries like this in your booklet, do take the time to highlight some of the keywords. So when I'm reading through this, I'm going to highlight some of those keywords I think I would need to use in my revision and in my examinations. So the first time you encounter a pathogen, your white blood cells detect it and either engulf the pathogen we've just learned about that, phagocytosis, or produce specific antibodies to destroy it. During the primary immune response, some lymphocytes, some of the ones that produce the antibodies, become what we call memory cells. And I'd say that is particularly key. That is what we're linking in with immunity here. So if we encounter the same pathogen again, we have a secondary immune response. The memory cells remember the pathogen, they've seen this before, and it means they can produce antibodies very, very quickly to destroy the pathogen before we can actually feel any symptoms. So we might not even know that pathogen has actually gone past our first line of defence. We have no idea. What we say is we become immune to this pathogen. We've got the right antibodies to fight it off. So looking at this in terms of two graphs. So on this first graph here, this primary immune response, on initial exposure, the non-specific immune response of phagocytosis and the specific immune response of lymphocytes will start to produce antibodies. This can take several days, though, to make the right antibodies, so you can start to feel quite unwell. So you can see the level of antibodies in the blood is quite low, and it also, you can see here, time in days, it takes over 10 days, close to 15 days, to make the maximum amount, not the maximum amount, but the peak amount of antibodies. So you can feel quite unwell in your first infection with this pathogen. However, if you look at the secondary immune response here, you can see the body has made memory lymphocytes that respond immediately to the pathogen and therefore create the right antibodies very quickly. So you can see the level of antibody in the blood is much, much, much higher. And as you can see by the gradient of this line, oh, sorry, I'm not very good with this. Sorry, my drawing. The gradient of this line is much steeper, so it produces the antibodies much quicker, and therefore we might not even show any of the symptoms of this pathogen in the first place. So this is the difference between our primary and our secondary immune response. So how can we use this in medical ways? And hopefully some of you have maybe started to already make the link talking about vaccinations, because a vaccination, we can actually cause this secondary immune response to be sort of ready. We can preempt it. We don't need to get infected by the live pathogen in the first place. So I'm sure you've all had some vaccinations before, and you maybe have heard of some of these vaccinations. And we're going to come on the end to talk about sort of vaccinations and things you might have seen in the news recently as well. So I'm just going to run over kind of the vaccination story, what happens. So a small quantity of dead or inactive weakened pathogen is injected into the body. You can talk about why that's important in your live lessons. The disease is weakened, so it's killed. That's really, really important because we don't want to make ourselves ill with primary immune response. So there we go. I'm just trying to show that it's being weakened or killed. It wouldn't really happen like that. The weakened or killed disease is given to the patient. So it's injected into the patient, into the blood. The white blood cells produce antibodies, as we've just spoken about. So these are our lymphocytes. They produce a range of antibodies. The right antibody is produced. So there we go. We can see it increasing in number. Sorry, my iPad's being a bit slow. The antibody, as we just learned, binds with the antigen on the surface of our pathogen, and the body can now produce the right antibodies, and it can start to fight the disease. Or it can fight off this pathogen and therefore fight the disease. I did say we'd come back to this. It's really important. People get confused between antibodies and antigens. It's crucial to keep this into your notes. So the antibody is the protein produced by our immune system. Antibody is a good guy, most of the time. So your antibody is the one that's trying to fight off the pathogen. The antigen is this guy here. The antigen is what is presented on the surface of the pathogen. That's why we can actually recognise it's foreign, it's not meant to be there, and the antibodies will bind to specific antigens. They've got to be the correct shape. So So just make sure you know the difference between the antigens and the antibodies. Antigens are the foreign ones, the ones that our body doesn't recognise. So how a vaccination works, you've got this summary table complete in your booklet, so do have a read over this. Vaccinations act as a reminder to the body of what antibodies they need to produce, like we've just run through. And you can also get booster vaccinations to refresh your lymphocytes' memory, to make sure it remembers those right antibodies that will bind with the antigen. Your task, I'm going to give your task. This will probably be given to you after your live lesson, but just a heads up, is to make a comic strip. I know there's not space for this necessarily in your booklet on OneNote, so you can do a separate page on OneNote. You'll ask your teachers, but I don't mind if you can do it through Word or any different app you'd like to use. But I'd like you to come up with a comic strip of how vaccinations work. Do make sure you include the keywords. So you want to be talking about antibodies and antigens, lymphocytes, pathogens, and at the end, do make sure you mention how it creates memory cells. So the whole purpose of this vaccination. You'll talk more about that task in your live lessons. Final thing to mention is herd immunity. And if some of you have already done some of the COVID reading that was set by Mr. Hartford, you will have come across this already. If a larger portion of the population is vaccinated against a pathogen, so not everybody, but the majority, it therefore stops the pathogen spreading. As you can see by this diagram here on herd immunity, so if I've got someone who's transmitting it, if they then give it to somebody who's immune, you can see we're blocked. Transmission is blocked. It's stopped in its tracks. However, if there's lots of people that are susceptible and we haven't been, a high proportion haven't been vaccinated against it, it can spread. So the pathogen, I should say, can spread. So the principle of herd immunity is if a larger proportion of your population is vaccinated, a pathogen cannot spread as easily because it won't be passed on from somebody who is immune. A few things you can look into, the MMR controversies, you can look into this. This is linking with both vaccination and herd immunity. So this might be something that you look into ahead of your live lesson for discussions during your live lesson. Some more questions. I'd pause the video now and have a quick read through these. Particularly the platinum question is quite a hard challenge question, and with some application looking at tuberculosis in cattle and badgers, thinking about different vectors there. This is something that I'll be discussing with my classes during the live lessons. And of course, probably a lot of you have been thinking this, but what about a vaccine for COVID? If you haven't already, from your teachers, do ask or they will share with you this brilliant resource made by Mr. Hartford, The Battle Against COVID-19, The Race for Immunity, Individual and Herd by Infection and Vaccination. And this picks up on a lot of the keywords we've been looking at today. It's sort of a 2-3 page document to read through, and it's something we'll definitely be discussing in live lessons this week because it's extremely topical. If you want to have a look at some more articles around this, I've given you a link here, and it's just talking about possible vaccinations for COVID, how long they would take, what the different vaccination avenues researchers are looking into at the moment as well. And it's a really interesting read. Some of it you might find quite challenging, but I think that's a really important thing to do at the moment. And don't worry if you don't understand this whole article. But definitely have a read of the one in yellow, and I'll be posting this on my class OneNote for my group to have a read through ahead of the live lesson. Good luck and see you in the live lesson.